This topic made me to little bit go into some research, and then I thought I can do some justice. So in this 8 minutes, I'll just cruise you through that. These are my disclosures. Once we understand the gut endocrinology, so basically the GI endocrinology is much more than digestion. It also includes absorption, gut motility, intestinal blood flow, and much more. And so these are mostly peptide in nature, and apart from the digestive system, it modulates so many other absorption and everything else, and it is modulated through the GI mucosa and the endocrine cells. So why actually are the intestines the largest endocrine organs? So if we go into the genomics, we see that there is a lot of structural homology. If you see this graph, we can see the black dots. The solid black dots are the hormones, right from which had been spanning from the decade of 1920s to the recent ones. The black dots are actually showing that these hormones bear certain amount of structural homology. Just the functions due to the carboxylation at the carboxyl end or the post-transcriptional and the translation, these have actually been imbibed from the same, like I would say, great-great-great-great-grandfather or mother, whatever, and it has the same structural homology. These act usually from the paracrine, that is the adjacent cells. The action can be adjacent and it can be also by the white expressions. So why this different gene expressions? And that the single hormone gene can be expressive of different bioactive peptides is because of the alternate splicing of the transcripts. The perfect examples are the secretin hormones, the calcitonin peptides. Also, it can be due to the multiple products of the prohormones, and it can also be due to the differential processing of the prohormones containing 2 or more active sequencing. So, when it comes to the extra-intestinal gene expression, see, one hormone can be expressed in tissues even outside the GI tract. For example, the cholecystokinin gene can be expressed in the cerebral and the peripheral neurons, the pituitary corticotrophs, the melanotrophs, the thyroid, the spermatogenic cells, and even the cardiac myocytes. So here are the so many means like the classification from the cholecystokinin, just taking the example of that, which has a multiple extra-intestinal gene expression. So one hormone can have so many manifestations all across the body. Then there are the cell-specific pro-hormone processing, that where the hormone goes post-translational processing, just like the gastrin hormone and the glucagon hormones. And just for example, gastrin hormone. This hormone can act in the antrum, the pancreas, the pituitary, and the colon, just because of the post-translational processing. Now coming to the GI hormones family, we are all very aware, nothing new. It is divided into the 3 major families, that is the gastrin family, the secretin family, and the others. I'll just brush through all these. The gastrin hormones are mostly found in the pyloric G cells, and the G17 component is the most active form of gastrin, and it acts, the main action is the gastric acid and the pepsin secretion. Also increases the gastric motility and the pancreatic secretion, the insulin secretion, and also the colonic contraction. The peptides and the amino acids and the abdominal distension, they are the major stimulating factors for this gastrin. And the somatostatin, the secretin, the GIP, and the vasoactive inhibitory peptide and the glucagon are the major inhibitory things for this gastrin. Coming to cholecystokinin, they are being seen in the L cells in the upper intestine, and they are found in the duodenum, jejunum, and also in the enteric and the pancreatic nerves and the brain. And it has a very short half-life. It basically, the action is to stimulate the pancreatic secretion in augmenting the secretin secretion and basically relaxing the sphincter of Oddi, and also causes the contraction of gallbladder and enhances the motility of small intestines. The secretin are found in the S cells, and it increases the pancreatic juice secretion and the bile acid secretion. It decreases the gastric acid secretion and the contraction of the pyloric sphincter. The gastric inhibitory peptide are found in the K cells of the duodenum and jejunum, and inhibits the gastric acid secretion and the motility and stimulates the insulin secretion. Coming to the vasoactive intestinal peptide, this decreases the GI motility, it causes vasodilatation, and the next is the somatostatin. It is basically a negative hormone. It causes the inhibition of so many hormones like gastrin, VIP, and even the insulin, but it decreases the insulin secretion. And it is found to precipitate the gallbladder stones and inhibits the absorption of glucose, amino acids, and triglycerides. Ghrelin is the first identified circulating hunger hormone produced by the P and D1 cells, mainly through the growth hormones receptor, and basically it is found the least when the patient or the person has taken the meal, and it is the maximum when the patient is in the starving state or the fasting state. And when we compare the leptin is the satiety hormone and the ghrelin is the hunger hormone. Other effects of ghrelin are that it decreases the gastric acid secretion and motility. Coming to the peptide YY, it inhibits the food intake and inhibits the gastric acid and the motility. Substance P, it is found in the endocrine and nerve cells of the GIT. It increases the intestinal motility of the small intestines. The neurotensin, it inhibits the gastric acid secretion but increases the ileal blood flow. The motilin increases GI motility. Now, once we have done through all these hormones, so what is the thing that we are actually learning from this presentation is the way forward, is the genomics and the microbiota that we are, that there are so many studies that have come up, the systemic reviews, meta-analysis, and also the biohacking. That we had a very good lecture yesterday by Dr. Vinay Khatri, and then there are, because of this understanding of these hormones and how they work, we have learned how they have actually, so many diseases are there and how the new diagnostic and therapeutic strategies can be combined to help the human. Then there are so many articles of ingredients and the GLP-1 and GIP, how it helps in treating the obesity and diabetes. Then it is all about the gut endocrinology and the biogenetics and the biohacking, I would say biotechnology. If we have to understand this and we have to go way forward, it is very, very imperative for all of us to understand this part, which we mostly tend to ignore as a surgeon, GI surgeon and a bariatric surgeon. Then the microbiota gut-brain axis. It's important that with the enhancement and involvement of the microbiota and the fecal transplantation, there are so many diseases that can be cured, like inflammatory bowel disease, the diabetes, that already has been published in our literature. To conclude, intestines have a diversified effects because of the hormones, and there is a robust control system required to coordinate the digestive process. And because if we understand this thing, and with the due course of time, it will definitely help understand the pathophysiology of obesity and other associated diseases, and it will definitely open the avenue for new diagnostic and therapeutic strategies. I thank Surendra Gale sir, who had designed this program and probably given this very tough, I would say, and I had to go through this topic. Thank you so much, sir.
Do we have any questions?